Welcome, everybody. Dick and I have had a little bit of a difficulty figuring what the heck we could possibly show that would get it across to everybody. Some of which are, doctors who are very familiar with this. Others are, like me, a computer scientist who knows nothing about this. And so, what I did and what we passed it off to each other was basically to say, let's try this approach of a 7-year-old, a presentation that would make sense to a 7-year-old, one that would make sense to a high school student, one that would make sense to a college student, and then one that would make sense to a PhD getting his PhD in genetics and immunization and stuff like that, and a doc, and then be at the same time. Okay, so this is what we're going to do today, and I wanted to give a little background on how this all works. On October 2nd, 2023, the Nobel Prize in Physiology or Medicine were announced, and Dr. Katalin, Karikó, and Drew Weissman of University of Pennsylvania.
Also of Lexington and Lexington High School.
Hey, don't steal my Okay, yes, Drew Weissman, and the reason we were very attractive is Drew Weissman went to LHS and then went on to Boston University, I believe, and then eventually ended up at Penn via Dr. Fauci's postdoc. Right. So there's an interesting path that he takes. And here you see pictures of them in one of the conferences that was held the day of the award.
Can I interject one thing?
Yes, please do.
At this point, I'd like to say that Lexington High School is really pretty good. Drew is the second person in 2 years to receive a Nobel Prize who went to Lexington High School. The year before was Carolyn Bertossi, and she got it in chemistry. Okay. And also I want to put a plug in for the high school because I've been a judge at the Lexington High School Science Fair for the last 10, 15 years. Some of you may have had students, or you yourselves, maybe you went through when you had AP chemistry, biology, and physics, you had to give a presentation at the science fair. That no longer is the case. So people who give presentations there now want to do it. This past year, there were 21 people who gave presentations. Of those 21, 13 of them received enough notoriety, whatever you want to call it. We judged them well. They went on to the regional science fair. At the regional science fair, one of the Lexington students shared first place. Another student shared second place. A third student shared third place at the regionals. From those regionals, 6 people have now been sent on to the state fair.
Wow.
Which goes on, I think it's April 4th. It's early April. So 6 people out of those 21 now are at the state fair. So Lexington High School does a pretty good job in science.
Very nice. And so the name or the work that the Nobel Prize was awarded for it was referred to as discoveries concerning the nucleoside base modifications that enabled the development of effective mRNA vaccines against COVID-19. It's a mouthful. And that indeed is the way the lectures, the last level of presentations are the language that they use. But the first ones don't use any of that, and it's a little easier to use. Another thing, the amount of time taken up by the videos we have uncovered exceeds the capacity of one of our meetings. So what we're going to do today, and I don't know what the next slide is, but let me just go ahead to it. Yeah, so it talks a little bit about what we did to create this, and it's this business about many different levels of explanation. So what we tried to do is give you a working knowledge of how it works without all the details of the biochemistry and chemistry and And also a lot of the sort of notion of how do you produce a vaccine efficiently for so that you can produce large quantities of it quickly. And that's really what Drew Weissman's work contributed. Oh, he did a lot of things, but they'll talk about that. But that issue of how do you, if you will, it's medical engineering, if you will. It's how do you take an idea that you produced in the lab successfully and then replicate it 3 billion or 13 billion times or something like that is how many doses they've given out. Okay, so the first two videos are something that's really nice. I love this guy's idea that he loves to explain complicated topics in plain old English. He's actually Belgian, I believe, and he does a really good job, and he has a whole bunch of videos on YouTube in this style. So you don't have to know everything, but you do get the gist of it. So here's the first one, and it's about basically how does it work. It doesn't really go into what, sort of what does it do as opposed to how it does it.
Developing vaccines is usually a very lengthy process, taking on average 8 years to get approved. In this video, I want to answer your questions about the mRNA vaccines for SARS-CoV-2 or COVID-19. Once again, I've listed all of my sources in the description, so please check them out if you have any doubts. Question 1. Is this video sponsored by the pharmaceutical industry? No, it wasn't sponsored by anyone, and in fact, so far, none of the Simply Explained videos have been sponsored. This one is no exception. If you want to help and keep this channel independent, consider becoming a member. Question 2. Why is a vaccine necessary? As this viewer points out, our immune system is quite capable. A vaccine is just a helping hand, strengthening our immune system so that fewer people need to be hospitalized and fewer people die. According to Johns Hopkins University, COVID-19 has an observed case fatality rate between 0.5 and 10%. Let's take 2% as the average and assume that everyone on Earth got infected. In this case, we would be looking at 154 million fatalities. We can largely prevent these huge losses by developing vaccines, so it makes a lot of sense to immunize as many people as we can. Question 3. Will the vaccine prevent the virus from spreading? That's not clear at the moment. After vaccination, your body will start making a specific type of antibody that will circulate in your bloodstream. However, the coronavirus enters our body mostly through our nose, and here we have different types of antibodies. It's currently not known how effective the vaccine's antibodies will be in this region. This means you won't get sick, but you could still have virus particles in your nose, ready to spread to others. That's also why vaccinated people can still test positive for the virus while not getting sick. But there is hope. One study exposed vaccinated primates to the virus and observed that they were able to clear the infection from their airways in a shorter time than others. This indicates that the vaccine reduced how long they were infectious. Time will tell if the same holds up for humans. Question number 4. How is the efficacy of a vaccine determined? The efficacy expresses the reduction of infections between vaccinated people and unvaccinated people. Let's take a real phase 3 study as an example. In this case, there were 36,523 participants, half of which got the vaccine, the other half got a placebo. Participants were closely monitored for 120 days while keeping track of COVID-19 infections. In the group that received the vaccine, 8 people eventually got COVID-19, while in the placebo group there was a total of 162 cases. The efficacy is then calculated by taking the difference between these groups and dividing them by the number of cases in the unvaccinated group. This gives us a 95% efficacy. This approach has been used many times before and is not new. Question 5. mRNA vaccines have never been approved before. They are unsafe and rushed. This is indeed the first time that mRNA vaccines are approved, but that's not because they were rushed or because corners were cut. First up, mRNA technology isn't new. It's been researched and worked on for more than 2 decades. It's only now that the technology has matured enough for widespread use. Secondly, we already studied other coronaviruses, such as SARS, in great detail. These have similar spikes, so we could build upon this knowledge. Thirdly, there was a lot of money available for research and production. The US alone invested over $10 billion into developing vaccines. This meant that companies could conduct clinical trials in parallel while also ramping up manufacturing. Here's how vaccines are traditionally developed. Notice how it's very sequential, one step at a time. And this is how the COVID-19 vaccines were developed. You can see that many steps were conducted in parallel, massively speeding up the process. Not skipping steps, but doing them at the same time. And finally, regulatory agencies such as the FDA and EMA gave priority to all studies related to COVID-19. Companies were allowed to periodically submit parts of their research for approval. This shortens the review time. So in summary, the vaccines were not rushed. They have been massively accelerated. Question 6. Why can't we use traditional vaccine technologies instead of mRNA? Well, we can and we do. There are many types of vaccines and mRNA is just one of them. An older, more commonly used technique is to use weakened viruses to boost your immune system. I won't go too much into details since this video is focused primarily on mRNA, but here is a handy overview of the types of vaccines and which company is working on them for COVID-19. Question 7. Can mRNA in the vaccine alter my DNA? No. Our DNA is contained in the nucleus of our cells. While mRNA does enter our cells, it cannot enter the cell nucleus. mRNA is naturally made by our body. When a cell needs a particular protein, the nucleus will make a string of mRNA containing instructions and give that to the rest of the cell for execution. So all we're doing with mRNA vaccines is temporarily giving our cells other instructions. Side fact, viruses that do become a part of us are called retroviruses. The most commonly known one is HIV. Question 8. What are adjuvants and are those dangerous? No, they are not dangerous. Adjuvants are used in almost all vaccines for the last 70 years. They signal your immune system to come check out what is happening near the place where you got injected with the vaccine. That way, the immune system will more quickly notice the odd spikes in your body, resulting in a more robust immune response with more antibodies. Question 9. Why do some people get sick from the vaccine? It's not uncommon for vaccines to have some side effects, and the ones for the coronavirus are no different. In this phase 3 study, side effects were common but mild. It included pain at the injection site, fever, tiredness, chills, and headaches. It also found that participants had more side effects from the second dose than the first dose, and that people older than 55 reported fewer side effects than younger participants. In summary, the mRNA vaccines are generally well tolerated. In most cases, having some mild side effects is better than actually having to fight off COVID-19. Question number 10. The coronavirus is mutating. Will the vaccine protect me from those mutations? We're talking about the UK and South African mutations, which allow the virus to spread more rapidly. Early studies indicate that the mRNA vaccines might be just as effective against the UK mutation. In this study, researchers took antibodies from people that had received the 2 doses of their vaccine and exposed them to the mutation. They found no reduction in neutralization activity. In other words, the mutated virus did not develop a resistance against the antibodies produced by the vaccine. The South African mutation is a different story. That same study found a significant reduction in neutralization activity, but they noted that the vaccine would still be effective. Some companies are already working on tweaked versions of their vaccines to offer full protection. However, we do have to be cautious with this data. It's an early study, so nothing conclusive just yet. So that was it for this video. All of the answers are published on my website, along with the sources and references that I've used. The link is in the description. Thanks for watching, consider subscribing, and stay healthy.
Okay, my apologies. I actually showed the second one first, but now let's go back to the first one that I intended to show. Let's see if I can get that to go.
You didn't say anything about the microchips that Bill Gates.
Well, that was 11. Yeah, I thought that was interesting because it was supposed to come after this one. And the idea being, I'll tell you how it works. Now I'll tell you why all those myths and concerns are indeed have reasonable answers. So here we go with the first one.
Developing vaccines is usually a very lengthy process, taking on average 8 years to get approved. Since a few years, we have the ability to create mRNA vaccines, which can be developed much faster. So how do these vaccines work? Are they safe, and how do they compare to traditional ones? But first you must understand how the immune system works. When a virus enters your body, it will attach itself to one of your cells and inject its DNA or RNA into it. This is like a blueprint for your cells, containing instructions on what the cell has to make. So in this case, the virus's RNA will tell your cell to make more copies of the same virus. They become factories, assembling new copies of the virus that can infect even more cells. But don't worry, our bodies have a defense system for foreign intruders. The immune system attacks any protein, virus, or bacteria that does not belong in our bodies, but it takes a few days for it to learn how to attack the intruder. Meanwhile, the virus factories are running non-stop, quickly replicating the virus and spreading it in your body. In other words, you start experiencing symptoms of whatever has infected you. After a few days, however, your immune system will have figured out how to attack the virus and will start producing antibodies. These attach themselves to the virus, preventing them from infecting more cells and marking them for destruction. As you can see, the immune system is remarkable, but it's slow to mount an attack. That is the reason why we can get sick in the first place. So to give it a helping hand, we developed vaccines. The main idea is to train your immune system to recognize and fight off an infection before it has occurred. Almost like showing your immune system a mugshot of the virus and saying, if you see this, kill it. There are various types of vaccines, but let's take a look at the new kid on the block, mRNA vaccines. To understand how they work, let's take the COVID-19 pandemic as an example. You might have seen pictures of the virus with its distinctive spikes. These spikes allow the virus to attach to specific cells in your body and infect them. Now, here's the key idea for the COVID-19 vaccine. What if we could train our immune system to recognize these spikes by having our own bodies produce them? To do that, researchers took the virus's blueprint, its RNA, and isolated the part responsible for producing the spikes. Armed with this blueprint, they created mRNA, or messenger RNA. This is a special form of RNA that can enter your cells and give them instructions. In this case, the RNA contains instructions to build the spikes of the coronavirus. Not the virus itself, just the spikes. So mRNA vaccines contain instructions for your cells that tell them to build a part of a virus in large volumes, almost like giving them a recipe to follow. Once this is happening, your immune system kicks into action and starts learning how to attack these intruders. Again, it takes some time for the immune system to fight off these spikes, but you won't get sick because it's only the spikes, not the virus itself. And that's it. Your immune system has learned how to attack the spikes of the coronavirus. It destroys all the spikes and even breaks down the mRNA vaccine itself. The only thing left in your body are special B cells or memory cells. These can linger around for months or years until the same virus infects you again. When that happens, the B cells can produce the correct antibodies right away, preventing the virus from spreading and making you sick. What's interesting about this mRNA technique is that it's relatively quick to develop a vaccine as soon as we know the DNA or RNA sequence of a virus. And secondly, because the vaccine only makes our bodies produce a tiny part of a virus, we cannot get sick from it. More traditional vaccines use weakened versions of the actual virus. This also triggers an immune response, but could also give you mild symptoms. Now you know how mRNA vaccines work, but don't take my word for it. All sources used to make this video are listed in the description below. Check it out if you want a more in-depth explanation. Give this video a thumbs up if you found it interesting and consider subscribing to the channel. Thanks for watching, until next time.
Okay,
Can I make one comment
Yeah,
on that
please.
before you go forward? There are a couple of things that I thought were misleading in that presentation. Excuse me. One is that he says that the immune system gets rid of the spikes. What happens is that the immune system recognizes the protein that made the spikes, and it gets rid of the whole virus. It doesn't just get rid of the spikes. only, which I thought it was a misconception I thought was in that side.
Yeah.
The other thing is what gets rid of the messenger RNA. That's typical, not typical. It is something the cells do all the time. They're constantly getting rid of messenger RNA. One of the major contributions to this particular Nobel Prize was by Karikó. And when we get to her part, you'll see what she did, taking advantage of or knowing that this is going on all the time. The messenger RNA is being destroyed all the time in the cells. And she overcame it with the messenger RNAs that she was. We'll get to that.
Okay. One other thing I would say about all of these videos is that I only really understood them after watching them several times. And so what I'm going to do is send out to our distribution list a link to this particular presentation, and you can go through it yourself at your leisure. But if you're still interested after this, I recommend that you do that, because that's really where you get the real understanding. Okay, let's see if I can figure out what I need to do next. Get on to the. All right. Get this. There we go. Okay. There's a bit of a delay. Okay. Okay. So the next video was actually made on October.
Before you move
Yes,
on,
go ahead.
Just
Yep.
a question. What confuses me a little bit is I know that a few months after I had, I think, my third vaccine, I got COVID. It was pretty bad for a few days. Now, if I hadn't had the vaccine, is the presumption that I would have had more virus particles and therefore would have been worse than I actually was?
Statistically, yes. If that answers the question.
Well, look at it this way. It's hard to know how sick you were, but it's easy to know hospitalization rates. And the hospitalization rates for people who've had the immunization are 1/20th of people who have not had hospitalization.
Okay, good enough.
So therefore, the extrapolation is that everybody is less sick.
Okay, thank you.
All right. So what this is a meeting that was held for the press the morning that the award was announced on October 2nd. And it was basically helping reporters then report about the award. And in particular, it has an excellent, the next level of explanation as to what the award was made for. So we'll start that one now.
Ladies and gentlemen, good morning and welcome to Nobel Forum for the announcement of this year's Nobel Prize in Physiology or Medicine. My name is Thomas Perlman and I'm the Secretary General of the Nobel Assembly. I will first read the announcement in Swedish, followed by English. We will then present the background to the prize and open up for questions. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2023 ska delas lika mellan Katalin, Karikó och Weissman för deras upptäckter rörande nukleosidbasmodifieringar som möjliggjorde utveckling av effektiva mRNA-vacciner mot covid-19. The Nobel Assembly at Karolinska Institutet has today decided to award the 2023 Nobel Prize in Physiology or Medicine jointly to Katalin Karikó and Weissman for their discoveries concerning nucleoside base modifications that enabled the development of effective mRNA vaccines against COVID-19. Here are the two laureates. Katalin Karikó was born in 1955 in Szolnok in Hungary. She performed her prize-winning studies at the University of Pennsylvania in Philadelphia in the United States. She is currently a professor at Szeged's University in Hungary and is also an adjunct professor at the University of Pennsylvania. Drew Weissman was born in 1959 in Lexington, Massachusetts, in the United States. He performed his prize-winning research together with Katalin Karikó at the University of Pennsylvania, where he's still active as a professor. I will now turn to Professor Richard Sandberg, member of the Nobel Committee, who will now describe the discovery. Please, Richard.
In today's interconnected society, the risk of new pandemics is greater than ever before. When the virus SARS-CoV-2 emerged in late 2019, it rapidly spread across the globe and developed into the devastating COVID-19 pandemic. During the viral infection, the immune system recognizes viral components as foreign, and they induce antiviral immune responses mediated through cells and antibodies. However, this takes time, and meanwhile, the person can develop serious disease. Vaccination strategies have been developed with the purpose of inducing antiviral immune responses that will protect against future infections. For a long time, weakened or inactivated viruses have been used as vaccines. Vaccination programs have been very successful in mitigating many life-threatening diseases, including polio, measles, and yellow fever. With advances in molecular biology, targeted vaccination strategies became feasible. By isolating genetic information from the virus, specific viral proteins could be produced and used as vaccines. Examples are highly effective vaccines against the hepatitis B virus and the human papillomavirus. More recently, harmless viral vectors have been used to deliver viral DNA fragments into human cells to make viral proteins and induce an immune response. Examples are the recently developed Ebola vaccines. The targeted strategies have the advantage of not requiring whole viruses, but still they rely on resource-intensive cell culturing for vaccine production. Researchers have therefore long been attracted to the idea of using messenger RNA, or mRNA, for clinical applications, where the mRNA is used as a template for protein synthesis. The production of mRNA does not require large-scale cell culturing, and future vaccines based on mRNA had the potential to become scalable, fast, and flexible. In fact, major advances were made in the last decades. Researchers showed already 40 years ago that large amounts of mRNA can be produced without cells, for example, using the T7 in vitro transcription system. Moreover, progress was being made in delivering mRNA into cells using different forms of lipid carriers. But there was a major obstacle to development of mRNA-based clinical applications. When in vitro produced mRNA was delivered into cells, it resulted in low protein amounts, and additionally, it triggered an inflammatory cytokine response. This year's Nobel laureates, Katalin Karikó and Drew Weissman, started working together at the University of Pennsylvania, bringing with them highly complementary expertise. Karikó had a background in RNA biochemistry and a strong vision of RNA-based therapeutics. Weissman had a background in immunology, working with dendritic cells and their role in immune surveillance and vaccination. It was clear to them that in vitro produced mRNA must somehow differ from the mRNA made in human cells. delivering in vitro produced mRNA into dendritic cells induced the inflammatory cytokine response, whereas the delivery of human mRNA did not. Using bacterial mRNA, shown on the right side, did also trigger the inflammatory response. Having explored several potential differences in the mRNA, they became interested in nucleoside base modifications. RNA contains 4 bases, often abbreviated as A, U, G, and C. However, these bases can get further chemically modified within our cells. It was becoming appreciated that base modifications were more prevalent in human RNA compared to RNA in bacteria. Could the lack of base modifications in mRNA produced in vitro be the critical factor that leads to inflammatory response? In a series of carefully constructed experiments, Karikó and Weissman used the T7 in vitro system to produce mRNA with various nucleoside base modifications. Each type of produced mRNA separately introduced into dendritic cells while monitoring the TNF-alpha levels, an important inflammatory cytokine. They discovered that mRNA produced with specific base modifications could almost completely abolish TNF-alpha secretion. In particular, modifications to the uridines, highlighted on the right side in orange, made a large difference. One of the most effective modified bases was pseudouridine, which is a naturally occurring uridine modification. Whereas in vitro produced mRNA triggered toll-like receptor activation, leading to inflammatory cytokines, mRNA made with pseudouridine did not. So Karikó and Weissman published their breakthrough discovery in 2005, 15 years before the COVID-19 pandemic. Realizing the importance of their discovery, they next demonstrated that base-modified mRNA resulted in higher protein expression in target cells, both in cell culture and in animals. More protein was made from each delivered mRNA molecule, and the laureates carefully dissected the cellular pathways involved. Karikó and Weissman made fundamental discoveries of the importance of base modifications in mRNA, which eliminated a major obstacle to mRNA-based clinical applications. Building upon this mRNA technology, as well as research by others on stabilized spike protein and mRNA delivery using lipid nanoparticles, 2 highly effective mRNA vaccines against COVID-19 were developed and approved in record time. Importantly, the 2 mRNA vaccines had complete substitution of uridines for a pseudouridine variant in the sequence coding for the spike protein. mRNA vaccines, together with other COVID-19 vaccines, have been administered over 13 billion times. Together, they have saved millions of lives, prevented severe COVID-19, reduced the overall disease burden, and enabled societies to open up again. The successful mRNA vaccines against COVID-19 have had a tremendous impact on the interest in mRNA-based technologies. mRNA technologies are now being used to develop vaccines against other infections. The technology may also be used for therapeutic protein delivery and cancer treatment in the hope of further improving human health. Here are the 2 Nobel laureates again. So this year's Nobel Prize recognizes their basic science discovery that fundamentally changed our understanding of how mRNA interacts with the immune system and had a major impact on society during the recent pandemic.
Thank you, Richard. I would like to open up for questions. we'll of course continue with interviews outside, but if there are any questions. Yes?
Have you managed to contact the winner? Who did you speak to, and what was their response to this announcement today?
Yeah, I was fortunately I was able to reach both of them, and they were very happy. It was wonderful to talk to them. This year I actually asked if they were surprised, because I suspected they maybe would not be having received many prizes, but Katalin Karikó explained that she was so overwhelmed, and also put it in context with her situation as a scientist for many years. 10 years ago, she told me that she was terminated from her current position and had to move to Germany for another position, and without her family and so forth. So it's been a dramatic change in her circumstances, and she acknowledged that, and she was great to talk to her and very happy. Drew Weissman as well. I mean, it was wonderful to talk to him, and he was overwhelmed and enormously grateful for this award. So yes, I got hold of them. Drew Weissman just a few minutes before coming down here. Yes.
Hello. Yeah, I'm Sufitin Aksasong from Green Post. I ask a maybe inconvenient question and need your further explanation. Do you really think that it's the vaccine and this COVID-19 pandemic? Thank you.
With this question, I would like to introduce our panel here. So I'll first introduce those who are sitting here. To the far right is Olle Kempe, Professor Olle Kempe, who's Vice Chair of the Nobel Committee. And then it's our Chair of the Nobel Committee, Professor Gunilla Carlsson-Hedestam, and I already introduced Professor Richard Sandberg. Perhaps, Gunilla, could you respond to that question?
Yes, it's of course a very reasonable question. As I think hasn't escaped anyone, this virus is still circulating, and there is now many variants that have spread, and this is of course how viruses behave. So once it has achieved this sort of level of spread, it's very difficult to stop it completely. But what's important here, I think, is that vaccines could be developed so fast, and this was, as we just heard, largely due to improvements in the technology and this basic discovery that allowed this. So I think in terms of saving lives, especially in the early phase of the pandemic, it was very important. Now most people have developed some level of immunity as a result of combined exposures to the virus and to multiple vaccinations. So we're not in, this situation is not as dire as it was a few years ago.
Any addition? Yes. Yeah. You'll get the microphone. It's coming here.
Thank you. This is Sunny Life Weekly from China. So my question is, considering the fast approval of mRNA-based vaccine against COVID-19, what impact can this year's prize have on the authorities to make decision in the future for mRNA-based vaccine development? And another question is, do you consider this year's prize as a powerful strike back for the anti-vaccine movement and also for vaccine hesitation?
If Olle Kempe, could you respond to those questions?
If I start with the latter part of your question, does this affect the anti-vaxxers and people who are hesitant? Anti-vaxxers, I don't know if they will change in any way, but I think giving a Nobel Prize for this COVID-19 vaccine may make hesitant people take the vaccine and be sure that it's very efficient and it's safe. Now we have 13 million tests being done all over the world on people who's not experienced the disease previously. And we know it's a very safe and very efficient vaccine.
And the other question was if it will, our decision to award, will influence authorities?
I think if the FDA and EMA will be faster, in a situation like this, I think they made the right decision to be very fast. In other cases, it may be wise to be a bit slower, I think.
Okay. Any additional question? Here's one, yes.
Hello, this is Caixin Media from China, and with the mRNA, we don't know the long-term effect yet. So do you have any concern over that?
Richard, would you like to respond to that question?
Yes. Of course, mRNA vaccines have only been given for a certain amount of time, which begs your question. Having said that, of course, the mRNA vaccine that is given is very transient, and the fact that 13 billion people have gotten the virus, vaccine virus, and the amount of side effects noted to date is extremely limited. So I don't think it's a major concern.
Anyone else want to add to that, or that's fine, I guess? I think that's.
Well, it can't be integrated into the nucleus, into the DNA, and that's, a safety precaution. The adverse effects that's been noted is mostly a mild myocarditis or perimyocarditis, mainly affecting young males, but that normally resolves without any long-term effects.
And maybe one could also add that the consequences of having COVID-19 can definitely have long-term health effects, as we know. So one always needs to relate the very minor effects of a vaccine to the severe risks of actually getting the disease. Yes?
Thank you, David Keaton, Associated Press. Obviously, throughout the pandemic, COVID-19 pandemic, we saw a very strong anti-vax movement, specifically targeting these new technology vaccines. As a scientific body, as scientists, what must this medical community do to try and breach this gap of trust between the wider audience, the wider public, and the science community to convince people that these are indeed safe?
Yeah, Gunilla, could you?
I think continuing to communicate how vaccines work, and for example, in the case of COVID-19, people felt that the vaccines were produced or developed very quickly. And that's true, because there was a huge need for that. But what these accelerated clinical trials did was not to remove any safety step. It was running things in parallel instead of sequentially. So there was no compromise in terms of the safety measures. And maybe that's not always understood. So, of course, communicating how it's done and all of the decades of basic research that's behind this kind of work, that's also not always appreciated by the public. But I think hopefully this prize can help highlight some of that, because this work has been going on since the 1990s.
Okay. Here is one more up here.
Hello, thank you. I'm from Chinese Nordic Time, and I'm happy. Actually, last year I'm sitting here and I asked the same question. So, well, mRNA vaccine will win the prize, but this year it gets a well-deserved prize. So I think everyone's happy with this. So the first question is about mRNA vaccine. So is there any other applications for mRNA vaccine in other diseases like cancer or like infectious diseases like this? So could you introduce a bit for the audience? Mm-hmm. The second one is about long COVID, because I think now long COVID impact is I mean, really important point for the society. So could you also let us know?
Maybe I'll ask Gunilla to answer the first part of the question and Olle Kempe the latter part.
Okay, so regarding other applications, yes, there's really quite a lot going on right now, and this is only going to increase most likely because we now have this increased safety data on the platform in general. But the main other applications I think we're going to see is, of course, many other vaccines against infections, particularly perhaps for flu, where we need updated vaccines. And that is one of the great advantages with this platform, is that it's flexible and fast, so one can update the vaccine if there's new virus variants coming, which we know happens all the time for flu. Other applications that are really sort of, there's going to be a lot of exciting findings, I think, coming out, and that's already started, is in terms of therapeutic cancer vaccines. So those are not preventative, but therapeutic vaccines, and they can either be done in a personalized manner or in a more general manner. Cancer is, of course, a very difficult disease, and it's in fact many, many different diseases, but cancer vaccines might very well benefit from this platform. And thirdly, there is many experiments and actually already clinical trials going on to deliver therapeutic proteins. And that can be replacing a defective enzyme, or you can be introducing an immunomodulatory protein. So in that sense, there is quite a lot of work that we will see, I think, apply to a variety of diseases.
So for the second part of your question, long COVID, well, first of all, vaccination prevents you from getting long COVID. Then long COVID is a spectrum of different diseases which ranging from the myocarditis that is usually much more prevalent if you get the COVID infection than from the vaccination. And it's also usually more severe. To long COVID, that is something we still don't understand the etiology of. And there is quite a lot of research ongoing on that.
Per. Varsågod. Ja, just det, du behöver en mick.
Yes, I'd like to ask you, Thomas, about Katalin Karikó's personal circumstances. You said that she had to leave her job, and could you just give me some more details about what happened to her and what the prize means now?
Yeah, I should say that I can only convey, and she essentially only told me what she has been telling in other interviews. And she's been explaining that her position, she never had a tenured position at University of Pennsylvania, and in 2013 it appeared as, from what she's saying, that it was hard to continue under those circumstances. She got a very senior position as vice president in a company, BioNTech, that later went on to make one of the vaccines. So she was not out of job in that way, but away from her family. She never received an R01 grant from NIH, which is a clear sign that she struggled and didn't get recognition for the importance of her vision. Because what was clear was that this is an extraordinary and unusual scientist who was very passionate about this idea of mRNA and using it therapeutically. And she resisted any temptation to sort of go away from that path and do something maybe easier, and maybe tell something about the funding situation, also being a female and being a foreigner and so forth. I'm sure it didn't make things easier, but she has not been complaining. She has also explained how happy she's been, and she was making progress through the years. So it was not that she was inactive, but she didn't get strong support, not the tenure, and the support to build up a big group. Instead, of course, she found very fruitful collaborations on the way. Yes. Is there anything more? I know several, or did, no, didn't see that. So, okay. So I think then, I know several are eager to have interviews. So I think we'll close this part of the press conference right now. Thank you so much for coming, and apologies for the short delay in the beginning. Thank you.
Okay. All right, that was certainly the beginning of that was, for me at least, difficult to understand, listen to it once. And but I think you get a sense as to what's involved. And that talk in particular is representative of the two Nobel lectures that are given that we'll see at some other time. But I think all of this discussion gives you prepared for the real scientific lecture that we'll show later on. But the next film is by Melissa Moore, who is involved in the production of the vaccine. She was the chief scientific officer of Moderna during the time when the COVID vaccine was produced. And this is a TED Talk, so it has the characteristics of TED Talks, which are pretty accessible. And she goes a lot further into the future in talking about what this means. So let me start up that presentation.
Now, many of the people watching this talk by now will have had 1, 2, 3, or even 4 doses of a messenger RNA vaccine. With billions of these shots now in arms, it's clear that this new way of making vaccines is both remarkably safe and incredibly effective. But did you know it's not the vaccine itself that's keeping you safe? It's actually you. Because the human body has amazing powers to both prevent and cure disease by making its own medicines. You just need to know what medicine to make. And that's what your vaccine gave you, simply a set of instructions for how to protect yourself against SARS-CoV-2. Now, vaccines are only the beginning. As said before, the advent of mRNA vaccines is heralding in an entirely new era of medicines. mRNAs give us the ability to not only prevent disease, but also treat previously intractable disorders. But before I get to that, let's talk about really what is this new way of medicines really about? Well, it all comes down to proteins. Now, you may think of protein as simply something that you need to eat, an important part of your diet, something that's important for you to build muscle. But it's not just muscle that contains protein. Protein makes up an a huge fraction of the incredibly complicated ecosystem that's your entire body. In many ways, your body functions like a large city, full of myriad buildings, interconnected buildings with lots of different structures. Now, just as the word building fails to capture the incredible variety of structures that make up any large city, the generic term protein gives no clue as to the incredible variety of molecular architectures at the molecular level. Like buildings, proteins are not monolithic. Our body makes many proteins, some like the collagen in our skin that makes our skin tough but pliable, like the actin and myosin in our muscles that enable us to move. Or our blood is full of hemoglobin ferrying oxygen around, antibodies protecting us from disease, and clotting factors that close up our wounds. And for any of you that have ever cracked a biochemistry textbook, this should look familiar. This is the metabolic chart of the human body. These are all of the reactions in your body that are keeping you alive and well right now. And each one of those reactions is catalyzed by a different protein whose names are shown here in blue. So proteins are not only what make your body, what make up the bulk of your body, they're also what make your body tick and keep you well. Now, if we think again about buildings, the structures of buildings may look quite different in the end, but they're all made of a limited set of building materials. And how those building materials, which of those building materials are used, and how they're arranged, and how they're attached to one another, gives the buildings their final form. The same is true of proteins. If we zoom down to the molecular level, we can see that proteins, if we unravel their three-dimensional architecture, are actually just long strings of building blocks. And these building blocks have different shapes and different propensities to interact with one another. So it's which building blocks are used and the order in which they are in the chain that gives a protein its three-dimensional shape. Now here I'm only showing 3 proteins, but creating and maintaining a healthy human requires the combined action of over 100,000 different types of protein. And our bodies make them all. Thus, our bodies are remarkable protein factories. At the molecular level, the numbers are truly mind-blowing. Each of the 30 trillion cells in your body, that's 3 with 13 zeros, contains between 1 and 10 billion protein molecules. That means that you have as many protein molecules in your body as there are stars in the known universe. Now, each different cell type in your body makes a different kind of protein, a different set of proteins, like the rods and cones in my eye that are detecting light right now, and the neurons in my brain that are interpreting that light and enabling me to see you right now. So they make a particular set of proteins unique to it. And like any complex building project, you can imagine that the process of protein synthesis needs to be tightly regulated so that the right protein is made at the right time and in the right place. But of course, with anything so complicated, it's perhaps not surprising that there's an occasional mistake, a fault in the algorithm. Let's go back to that metabolic chart. It's estimated that 1 in 1,000 newborns are born without the ability to make one of the proteins on this chart. Therefore, they have lifelong complications due to inborn metabolic errors. Let's take just one of those. Let's talk about von Gerke's disease, or glycogen storage disease 1. This is due to the lack of a protein circled here in red, whose job it is to release stored sugars so that you can maintain a healthy blood sugar level while you're fasting. So von Gerke's disease patients can't fast. They must constantly eat small amounts of carbohydrates, including getting up every 1 or 2 hours during the night to eat raw cornstarch. Now imagine the toll that this takes on parents. If they are ever to miss a feeding of their child could slip into severe hypoglycemia, seizures, and possibly death. But even if these patients can keep up this endless feeding cycle, they are plagued by lifelong complications, including delayed puberty, frequent infections, kidney disease, and liver cancer. So von Gerke's disease is just one example of a disorder where we know what protein is missing. What if we could give those patients back the ability to make that missing protein? Then we could actually treat their disease instead of just managing their symptoms. And that's where mRNA comes in. That's also where I come in. You see, I spent the better part of my career as an academic doing curiosity-based research into the fundamental principles of how proteins are made. And my specialty was messenger RNA. Like proteins, messenger RNAs are long chain-like molecules composed of building blocks. The 4 building blocks that make up messenger RNAs form what is known as the genetic code. As their name implies, messenger RNAs carry messages, messages that are translated by your body in order to create proteins. Thus, messenger RNAs are the language of life. And the human body has a lot to say. So, like proteins, your cells are chock full of messenger RNA. Every one of your 30 trillion cells has hundreds of thousands of messenger RNA molecules. Messenger RNAs are an essential component of all living organisms. So when you are eating protein-rich foods, you're not only eating protein, you're also eating lots of messenger RNA. Your body takes the messenger RNA in the food that you consumed, breaks it down into those component parts, and then builds new messenger RNAs specific to your needs. Now this continual destruction and rebuilding is a feature true of almost all proteins and messenger RNAs in your body. Let's take, for example, the circadian clock. This is the timer in your body that tells you when to be active and when to sleep. The proteins that make up this clock appear and disappear with remarkable regularity every day. The way that this is accomplished is that your body makes the messenger RNAs that encode those proteins appear and disappear every day. Every day for your entire life, you get your daily dose of clock messenger RNAs producing clock proteins. Now, three properties of proper medicines are that their effects are of limited duration, that their effects are dose-dependent, and that they can be given over and over again to produce the same effect. mRNAs are transient. The amount of protein produced is dependent on how much of that mRNA is present, and they can be induced over and over again to produce the same effect. So wow, it seems so simple. If we could treat a disease, if there's a protein that's missing to treat a disease, then we could simply give a few copies of an mRNA to the body for it to produce that protein. If that protein's only needed once, then maybe a single dose would suffice. If a protein is needed multiple times, then we can dose mRNA over and over again. And that's exactly what's happening. So when I went on clinicaltrials.gov this morning, it turns out that there are over 175 clinical trials now open using mRNA-based medicines that are recruiting patients. Another 54 clinical trials are waiting in the wings, ready to be opened. So there is a coming tsunami of mRNA medicines. Last year, Moderna and AstraZeneca reported positive results from a clinical trial where patients who, during open heart surgery, were dosed with messenger RNA injected directly into their heart muscles that told their heart muscles to grow new blood vessels in order to get around clogged arteries. In other clinical trials, we are repeatedly dosing patients with inborn metabolic errors to treat their metabolic disease. In fact, one of those clinical trials that's currently recruiting patients is for von Gerke's disease. And for cancer patients, we're creating personalized cancer vaccines. These vaccines are meant to train their bodies, their immune systems, to attack their cancers. These are truly personalized medicines, one vaccine for one person. Now, for personalized cancer vaccines to be the most effective, we need to get them made and back to the patient as quickly as possible. We aim for a turnaround time of 45 days. By January of 2020, we had already manufactured, quality controlled, and delivered to several dozen patients personalized cancer vaccines. So we had the know-how and the capacity to manufacture vaccines quickly. Thus, when the sequence of the SARS-CoV-2 virus was posted to a public web server on January 10th, 2020, we got immediately to work. Within 2 days, we had agreed with our collaborators at NIH on exactly which form of the spike protein to put in our vaccine. Because we had done so many times before, it then took our mRNA design team just 1 hour to design the mRNA that we immediately put onto our manufacturing equipment. We were then able to make that RNA, get it quality controlled, fill finish, and shipped off to NIH for the clinical trial in 45 days. Now, what I find truly remarkable is that mRNA sequence that took us 1 hour to design is the same mRNA sequence that went into your arms, that ended up in Spikevax, our now fully approved vaccine. 1 hour to design a medicine that has saved countless lives. It still gives me goosebumps every time I talk about it. So what does the future hold? Well, I've already told you about regenerative medicine and personalized cancer vaccines. For cancer patients, we can also send in, by directly injecting messenger RNA into their tumors, we can send in instructions telling the tumor cells to self-destruct. Or having the tumor cells send out signals to the immune system, to beckoning the immune system to attack. For patients with autoimmune disorders, we can send in signals that tamp down their overactive immune systems. And we and others are rapidly making many more messenger RNA vaccines. Because messenger RNA vaccines can be produced so quickly and rapidly, they're really well suited for newly emerging diseases, as well as other viruses like the flu, where new variants come out every year and the vaccines need to be updated. But one of the exciting things about mRNA medicines is we're not limited to sending in the instructions for one protein at a time. mRNA medicines can be easily multiplexed. Therefore, we're working on a combination vaccine for COVID, flu, and respiratory syncytial virus, or RSV, all leading causes of hospitalization and death in the elderly. And we're hoping that this will then be an annual booster that you will get just like the flu vaccine. So finally, the very modest footprint of the manufacturing equipment for making messenger RNAs means that they can be made almost anywhere in the world. And to take this to an extreme, the American Defense Department started a program in 2019, and we're working with them to miniaturize the entire process so that it can be fit into a single shipping container for rapid deployment anywhere in the world. So to finish, I hope I've convinced you that we have entered an entirely new era of medicine. Having learned to speak the language of mRNA, the language of life, we can now use it to create medicines that are just for one person, like a personalized cancer vaccine, or can be rapidly produced and distributed to entire populations like the COVID-19 vaccines. And the best part is we're simply tapping into your body's own ability to make its own medicines. Thank you.
We're going to stop here today, but I want to tell you that we do have in the wings. The two separate lectures that the two Nobel Prize awardees gave, they're each about 45 minutes long. So I'm not going to try and show them today.